You know, I have to unget
Okay.
my laser pointer out. Oh, goodness.
Oh, to get rid of that window.
I can't get rid of it. Oh, sure. Great. Love it. Glad this was all recorded.
I'll edit.
Great. Okay, I think now we're okay. Does that seem right?
I think so. Okay, well, welcome to today's astrobiology seminar. And today's speaker is Sarah Maurer from Central Connecticut State University, where she's been a professor for, well, since 2013. And she did her PhD at UC Santa Cruz, and then a postdoc at the University of Akron in Ohio. And she does research on trying to figure out lifelike functions in models of cells and protocells. And she's going to tell us about some of the work she's been doing about protocells. So take it away.
Thanks. All right. So I am located in Central Connecticut. We're just outside of Hartford. We are a regional undergraduate institution. So all of the work you see will have been done by and a single master student that I have borrowed from the Department of Biomolecular Science.
Hi, Bear. She's Bear.
Please
mute yourself.
And so there are some advantages to working with undergraduates, but there are obviously some disadvantages too. They have very short time span in my lab, so they usually graduate after a year or two of work. Sometimes they only do work during the semester, so they don't have time during the summer, or they go and do REUs at bigger institutions. And so we actually lose our best students through the summer. And if you're interested in talking more about what it's like to do research at a PUI, I would love to talk to you more about that after this talk. So the research that I'm going to present today is some of the more recent work that I've been doing. We synthesize amino acid decyl esters and then we characterize them. I'm also going to be talking a little bit about some of my work on complex prebiotic mixtures and some of the other projects that I have my hands in. It turns out that the other advantage of working with undergraduates is you can pilot lots of little kind of projects and see how they pan out. And if you really like them, then you can write a bigger grant. And it is really pretty low intensity. And so I do tend to have a wide variety of projects that I've worked on across prebiotic chemistry. And just to show you a little bit about this image is a micrograph of vesicles. And these vesicles are formed from our amino acid decyl esters. The brightness is from a fluorescent dye, Nile red, that embeds itself in our membranes. And it's not red because this is a monochromatic camera. You can also see that some of these show vesicles inside of other vesicles. This is really common. These are multi-layered. Even this one really bright ring is probably a few layers thick to get that really intense brightness. So we spend a lot of time doing microscopy, but we also do analytical chemistry in my lab. Before we actually talk about some of the chemistry that I've been doing, I wanted to talk a little bit about astrobiology, inclusivity, and a case for what I call descriptive naming. And so when you look at these images that are on the board, for example, this image that's all the way on the right, what do you call this type of interaction? Participation from the audience. No one knows what this is called. No chemists in the room. What kind of base pairing?
Hydrogen bond.
I think that most of us would call this Watson and Crick base pairing. Well, at least the biochemist in me does. And that's what I teach in class. Likewise, when you look at this diagram, what would you usually call this reaction? You would call it Miller-Urey. It's the Miller-Urey reaction. And similarly, this diagram in the middle, a lot of times we'd call this evolution, but we tend to call it a specific type of evolution. Darwinian, right? And so we use these capital N names to kind of obscure the fact of what they actually are. And this isn't intentional. Essentially, when you publish a paper, if I'm talking to Roy, I say, hey, did you see the new Rajamani paper? Because we both know her and we know her work and we're going to talk about her new paper, right? And so it comes from that. We use these capital names because there are seminal papers that describe some new concept or theory. And then we end up using that consistently as we grow as scientists. But what it means for the astrobiology community is that we end up with a bunch of jargon. So I think a really good example of this is that most of you didn't automatically think of Watson and Crick base pairing, right? You think of that as like, oh, that's like the normal type of base pairing that we see in DNA, or like, oh yeah, that's like an electrostatic interaction. And you don't really think about the people behind it. And so when you're teaching a class, or when you're talking to a grad student who's new to the field, or maybe from outside your field, and you use terms like Miller-Urey, what you're really doing is quizzing them on how well they know the background. And so what I am advocating for today is that instead of using those capital N names when we're teaching classes and when we are talking to our peers and our students and when we're writing grants, that instead we use more descriptive, more inclusive naming. And so in this case, if you're talking about the most normal type of base pairing, calling it standard base pairing is a much more effective way of communicating the idea that, oh, this is the typical way that DNA interacts with itself, instead of the non-standard base pairing, which is like the fanfic of base pairing. And that would be Hoogsteen base pairing. So I'm sure you all knew that. And so just as you're moving through the astrobiology community, I would encourage you to think about moving away from jargon and moving towards more descriptive approaches to naming your science. All right, that was my PSA for the day. So now let's talk about what I actually work on, which is thinking about lipids, how they form membranes, and how those membranes can lead to lifelike functions. And so when we think about a top-down approach to astrobiology, we think about the biological approach that organisms use today, and we have lots of membrane-forming lipids. So in the chemical structure that you see here, we call these long carbon tails here, the hydrophobic tail, and this is what forms the hydrophobic barrier of our membrane. And then we have a glycerol backbone, these oxygens here, and they are linked covalently to a phosphate. This phosphate then is covalently linked to some R group, and that R group determines what we call the phospholipid. And so there are a large number of phospholipids that are used across humans and bacteria, and you can see their chemical structure here. The thing that we care about a lot in biochemistry is not just how hydrophobic something is, but also how hydrophilic something is. So hydrophilic means water loving, and things with a charge tend to interact really well with water. And so you can see that in phospholipids, that phosphate group is negatively charged and is going to interact with water pretty well. Likewise, a lot of these lipid head groups, like the phosphatidylethanol amine, the second head group down, has a primary amine on it. And so we get a head group that is what we call zwitterionic. It both has a negative charge and a positive charge. And this makes it really, really good at interacting with water. And you can see that in these head groups, we have some positive charges. Phosphatidylserine has both a negative charge and a positive charge. And then there's some head groups that have no additional charge. And additionally, there are some that don't have phosphate. So for example, in the glycolipid group, that would be up to this oxygen, and then we attach a sugar, which is just polar. So you can have some different head group types that like to interact with water. Sugar, you know, dissolves in water. It interacts very well with water. And so when you put a sugar head group on our lipids, it means that it's going to form a fairly hydrophilic head group. You'll note that in this slide, I'm only showing the head groups for our eukaryotic and bacterial cells. It turns out that archaea have very different membrane forming lipids, like so different that when we first found out about them, we didn't recognize what they were. So if we look at the bacterial lipids, those are in orange here, and archaeal lipids are in blue. And you can see, first of all, that instead of having this fatty acid tail, these are branched. Isoprenoids are 5 carbon units. Fatty acids are made from acetyl-CoA, which are 2 carbon units. So in our bacterial lipids and eukaryotic lipids, we have all even chained fatty acids, but in isoprenoids, they're 5 carbons. So there's a 5 carbon, a 10 carbon, a 15 carbon, a 20, right? And so it's very different in the way that it's synthesized from a hydrophobic standpoint. Instead of an ester linkage to the glycerol backbone, they are ether linked. This tends to make them a little more stable. Ester bonds hydrolyze with water pretty easily. And so the ether linkage means that archaeal membranes have less likely to hydrolyze in water. And then additionally, they are oppositely chiral. Their handedness is different, which means that the enzymes that are used to synthesize them must be very different active sites, because you have to stick the phosphate on to kind of the opposite side. And in this picture, they actually kind of look pretty similar. I'm just going to go back one and remind you, this is what a phospholipid looks like. They all kind of look like this, 2 tails, glycerol head group. But archaeal lipids actually look like this. So they are very, very odd. Not only do you get this standard 2-tailed lipid, but you also have what are called tetraether lipids. So the tetraether lipids they have 2 head groups. They are membrane spanning. Oftentimes, one head group is different than the other, so they're not going to have a phosphate on one end and they'll have a phosphate on one end and a sugar on the other. Here in these structures, we're not actually showing head groups, but archaeal lipids often have really large sugar head groups. Sometimes they're di, tri, or tetra saccharides, so kind of like a sucrose or even bigger, attached to those head groups. And additionally, in addition to being branched, they also have cyclic alkanes in their tails. And so one could, from all of this, surmise that the number of membrane-forming lipids that are available to biology today is much greater in scope than when you think about something like DNA, which has 2 backbone types and 5 nucleobases, right? Here we're talking about the molecular diversity of the class of membrane-forming lipids is just a very, very large class of compounds. And you could imagine that part of the reason for this is that membranes have to have a wide variety of protections against environmental factors. They're not protected from the environment like nucleic acids are inside of that nice warm cytosol. That membranes have a broader distribution of functions, right? They have to not only transduce energy, but they have to signal other cells. There's a lot of protection, but they have to let stuff out and let stuff in. And so maybe we need greater diversity of lipids to achieve all of those various functions. And so the reason I'm telling you about all of this is because I think that a top-down approach can also inform our understanding of prebiotic lipids and what we need to form a stable prebiotic membrane. All right, let's just kind of switch gears. So knowing all of that background information on modern lipids, some of the questions that I have about prebiotic chemistry and protocells is, what does our prebiotic inventory look like for amphiphilic compounds? And then also, what kind of conditions and environments and mixtures of prebiotic amphiphiles form the most stable membranes, and how can they be used? So when we think about these realistic prebiotic inventories, a lot of times we think about these high energy reactions producing abiotic organic molecules. And this can be interstellar synthesis that's delivered to Earth by meteorite delivery, or this can be from terrestrial synthesis, geothermal activity, spark discharge, volcanic activity, and however else you want to make your organic compounds 4 billion years ago on Earth. We have some idea of the prebiotic inventory for amphiphiles. And one of the first proposed, we're going to dig into history a little bit. One of the first proposed prebiotic amphiphiles is a fatty acid called decanoic acid. And so you can see that this was published in 1978 by my PhD advisor. I was Dave's last graduate student. And decanoic acid in this paper was shown to, when you put it in water and heat it up a little bit, it forms an oil phase. And that's because the carboxylic acid, when it's protonated, is not very amphiphilic. That protonation doesn't give it enough ability to interact with water. And so it literally looks like an oil droplet floating on the top of your test tube on a layer of water. If you take that oil and you mix it with sodium hydroxide, this proton is going to react with the OH here and form water. And you get a negatively charged decanoic acid, which we call decanoate. That negative charge allows it to interact with water really well. If you deprotonate all of your decanoic acid, you end up with a solution that's called a micelle solution. And those micelles are so small that you cannot see them with your eye. So they tend to be less than 100 nanometers. You need 400 nanometer visible light to scatter. And so micelles are essentially, they look like a clear solution, but they are nano aggregates in solution. Now, if you add HCl to that solution and you bring the sample back to pH 7, you get vesicles to form. And it's really, really great. The vesicles from decanoic acid are very obvious under the microscope. They're very easy to see. But the interesting thing about this research is that if you take the oil to pH 7 using NaOH, no vesicles form, none. And when I was doing undergraduate research, when I was doing graduate work, I didn't think this made any sense, right? Like, there's no reason that you should have to go to a micelle state to make vesicles. And so during my first few years as a PI, I realized that what happens when you mix sodium hydroxide with hydrochloric acid, you get sodium chloride and H2O. And it turns out that if you take your oil and you pH adjust it and you add salt, you get vesicles to form. And so it is very obvious from everything I just told you that fatty acids are a little bit overrated as prebiotic membranes because the environment is so constraining to make vesicles. I already told you that they're temperature sensitive, right? You need to melt that oil. So decanoic acid melts at 27, but if you increase the temperature to say 80 degrees, all of the decanoic acid dissolves into the water phase. It's pretty soluble and you don't get any vesicles. It is dependent on the ionic strength of your solution. Sodium chloride is good in small amounts, but in huge amounts, it causes kind of vesicle collapse. Additionally, divalent cations are insoluble in the presence of decanoate, which is why soap scum exists. And so they precipitate in the presence of magnesium and calcium and iron 2+. And then on top of that, they work only in a narrow pH range. So you have to work with them between pH 6.8 and 7.4. And contaminants tend to make them less good at forming membranes. It turns out that sometimes contaminants make them better. And so maybe messier mixtures are good with this kind of system. And additionally, the negative charge can assemble on surfaces. And so sometimes you get all of your decanoate precipitating out because it's associating with like a mineral surface. And Fatty acids are a great model because they are so cheap. Like a jar of decanoic acid, I can like give it to every undergrad that comes in my lab and I wouldn't run out of money. But they, and they're pretty easy to form membranes. It's not a dangerous process, but they have a very limited constrained usage. And I think from my introduction on lipids, it has kind of become obvious that there's a bigger inventory of possible molecules that can form membranes, and constraining ourselves to just using fatty acids as a model may not be the best subset of molecules for prebiotic vesicles. Some of the ones that are formed through like meteoritic synthesis are things like decanol. And decanol on its own doesn't form membranes, but it definitely acts as a co-surfactant in most membrane forming systems. Similarly, you can get branched chain isoprenes that have an alcohol head group, so looking more like the archaeal version of a prebiotic amphiphile. You can also get amphiphilic molecules through dry downs, so you can condense a glycerol on a fatty acid. You can also condense a phosphate on an alcohol and get a phosphatic decyl phosphate. And then there are some reactions that are combinations. So this molecule is kind of like our glycerol molecule plus a phosphate. And then there's this really strange class of molecules, these keto acids that are a pyruvate derivative that are formed through UV light. So you can actually cause them to form through a UV synthesis. And so there's a pretty broad range of prebiotic molecules. But if you think back to our phospholipids, one thing that you may notice is that there are no positively charged molecules in this prebiotic inventory. And I think that positively charged molecules are very important to modern cells because having the positive charge allows them to interact with different molecules. It modulates their surface charge. And so one of the ways that I have shown that you can get positively charged molecules is just by condensing an amino acid onto an alcohol. To do this, you take your amino acid and you put it in a 1-to-1 mole ratio with your alcohol. Here I'm using alanine and decanol. And I have it at low pH, and you can tell that because you're all biochemists who know that when a carboxylic acid is protonated, the pH must be below pH 2. And usually between 2 and 3 is fine. There are sufficient protonations to drive the reaction. And when we have this at low pH in equimolar concentrations, if we leave it in the oven over the weekend and come back on Monday and rehydrate the solution, what we see is in the mass spec, we see the exact mass of our product. So this is what I call a decyl alanine ester. And it has a mass of 230 grams per mole. We also see that in that reaction sample, it has now started to produce membranes. So alanine doesn't form membranes and decanol doesn't form membranes, but through this dry down reaction, we really reliably get membranes to form, and in great abundance. You can dilute these solutions tenfold and they still form membranes. And so we are producing a pretty significant amount of product. Here it says 50% yield. I think if we optimize the time that it is in the oven and the temperature, that we could get more than 50% yield on these reactions. And we're not purifying anything. So I am just taking my dry down and bringing it up in solution. This is really very, very easy to get to function in the lab, which is great for undergrads. The reason that I really like alcohols and I really don't like fatty acids, in addition to fatty acids being somewhat hard to make form vesicles, alcohols seem to be more abundant from some of our prebiotic syntheses. So this is the common type of synthesis that you see on meteorites. And of course, it somewhat depends on the gas inputs that you use for your synthesis. But you can see that our alcohol, so something like decanol, is the highest abundant molecule in this reaction. There are some alkanes, like this would be something like octane, right? Like gasoline octane, that it doesn't form membranes at all. It forms an oil phase. And then our fatty acids are the N-alkanoic acids, and they are in somewhat lower abundance. And so we would expect alcohols to be present on early Earth, but also we'd expect them to be present in considerable abundance. Similarly, we like amino acids because amino acids are present in meteorite samples. We also know that they're present in spark discharge. They kind of show up everywhere that you do high energy radiation of nitrogen, carbon dioxide. They just kind of are a product of that. That's me hand waving it. And so we like both of these molecules. We think they were abundant on early Earth. And so not just alanine, but a large number of amino acids form products. So everything listed here, we saw exact mass in the mass spec, meaning some product had formed, although some of the molecules seem to form less product than others. That's very qualitative, it's not quantitative. We also tried some non-biological amino acids. They form product. Some of them form vesicles, some of them do not. The ones that have stars next to them had so much product, it was in such abundance that to see vesicles, we actually had to dilute the sample because it was forming like a gel phase at the amount that we were synthesizing it at. Lots of the amino acids that form membranes are in what we call the prebiotic set. When I teach biochemistry, I usually call those the boring amino acids. So things like glycine, leucine, valine, they have aliphatic side chains. They're not very chemically interesting, but they are often found in prebiotic synthesis. Also, aspartic acid and glutamic acid are sometimes considered prebiotic, depending on who you ask. And those form vesicles. Really interestingly are aromatics. So phenylalanine, tyrosine, tryptophan, they do not form vesicles. They form product. So we see product in our mass spec, but we don't see structures forming. So the aggregation doesn't really happen. Some of them are very chunky. Phenylalanine is very poorly behaved. And then our positively charged amino acids, those are arginine, histidine, and lysine, also don't form membranes as decyl esters. And so this is kind of an interesting result. All of these samples were done in binary mixtures. So 1 millimole of decanol with 1 millimole of amino acid reacted. So this is not messy chemistry at all. This is very tractable chemistry that we can analyze analytically. It does seem that like when we go to 80 degrees, we see less product. And when we go to 100 degrees, we see almost no product from this synthesis. Things turn very, very brown very quickly. All right, so one of the things that we have tried to do more recently, so all of that work has been published, but this is unpublished work. And more recently, we wanted to make the reaction sample more complex. So sure, in a one-to-one binary mixture, you get product, but if you have a bunch of amino acids together and a bunch of alcohols, like you would see from Fischer-Tropsch synthesis or a meteorite synthesis, do you get products, right? Like that's the big question. We tried that. We took a bunch of amino acids and a bunch of alcohols, all the alcohols that I could find in our department that were normal chain. We mixed them together, did a dry down, and our mass spec was broken. So we don't really, I mean, at that point, our mass spec was broken. So I don't know if we formed product, but I know we didn't form any membranes. There were no membranes in that sample. I like to do the hard experiment first and just see if it works, because if it does work, then like you don't have to do all those little tiny incremental experiments. But when it didn't work, we decided to try a more tractable mixture. And so here we are using alanine, proline, octanol, and decanol is our complex mixture. And we chose these specifically because octanol, we know, is a little worse at forming membranes than decanol. And alanine is like the superstar of this. It just is really, really good at forming membranes from this decyl ester. Whereas proline is like a boring amino acid, but it's not very good at forming, like we could tell that the vesicles were not as abundant. And so we kind of tried to pick things that were on the edge of stability so that we could get more information. If everything forms membranes all the time, you don't get very much information out of it, right? You want something that's kind of bad at its job. And so to do real workup of this, we wanted to analyze yield as well as analyze morphology. So to do yields, we used our LC that's coupled to a UV detector and a mass spec. And in this detection, we are using very low UV. So in analytical chemistry, you should know that kind of everything absorbs in the low UV. Methanol has some absorbance in the low UV. Our acetonitrile, which we're using to run our samples through, has some absorbance in the low UV. And so it's really good because it detects literally everything, but also it's finicky because you detect everything. And so in this trace, our ester products lump at the front of our retention time, and our two amino acids, proline and alanine, come out at a later retention time. And when you're doing UV at 210, you can somewhat approximate the peak area to the concentration. So bigger peak area is going to be more abundant, smaller peak area, less abundant products. We also can do mass spec on these and get exact mass traces. I'm going to show you that. They're a little more messy. They don't have such a nice peak. And these are our 4 products. So here we have, oh, this is total ion count. This is our alanine decyl ester. This is our proline octyl ester, our alanine octyl ester, and our proline decyl ester. And so we know that they're all coming out in that range. We also can't use these traces to quantify because alanine is not as good at ionizing in a mass spec as proline. So you can't really compare peak areas between mass spec traces just because they're so bad at being quantitative. Also, there are no standards available for these molecules. And so I can't like buy a standard and equate it. All right. So just to show you what that quantification looks like, and to walk you through this reminder, we have our alanine with octanol, our proline with octanol, and a mixture of alanine and proline with octanol. Those are the first 3 samples here. And in this, every reactant is a 1 millimole amount. And so it's 1 millimole of alanine reacting with 1 millimole of octanol. And what we see is that the octanol products seem to be less abundant than our decanol products. And here I'm showing you samples in duplicate so you can kind of get an idea for the deviation between samples. And so when we look at our octanol containing samples, they seem to be slightly less abundant than our decanol containing samples, although maybe not statistically less abundant. But we do see that when we have both alanine and decanol present, that we do get more abundant products. And so this is telling you that when you double the amount of alcohol, you are getting more product out. Interestingly, when you double the amount of amino acid, you do not get more product out. We are not seeing an abundance of product in the double amino acid. So it seems like the alcohol concentration is somewhat controlling the amount of product that we make. The other kind of observation from this is that it seems like the proline products are slightly more abundant. And we have this one major outlier. And so obviously more replications are needed before publication. But essentially, this gives us a pretty good idea of how product is forming. The other thing that we looked at is morphology. And so when you're looking at morphology, just a general understanding of, is it turbid or is it not? So if you see cloudiness, that means that something's aggregating. And if it looks clear, then that means nothing is aggregating. So for example, in this sample, you can see the reflection of my ceiling light very, very well, meaning this is not a cloudy sample. The ceiling light's reflecting off the bottom of the test tube, and there's nothing impairing that light coming through the sample. Whereas in this sample, you can see that this is slightly less 1 green, and also the ceiling light is less visible. And so that indicates that it is more turbid. There's more light scattering. That means that there's more stuff in solution. And so interestingly, and I told you this, alanine we know is a really good membrane former with decanol. And so our alanine octanol sample is slightly turbid. With proline, clear. Alanine plus proline, also clear. I don't know if there are micelles in these solutions, but we know there's product. If you look at the alanine decanol, very cloudy. If you look at the proline decanol, it is definitely cloudy, although maybe not as cloudy as the alanine decanol. Like I said, it's just not as good at forming membranes, but it is definitely not clear like the proline octanol. And then really interestingly, if you have alanine and proline in a reaction sample with decanol, the product is clear. And this is non-predictive, right? Like you wouldn't think that doing the reaction with both alanine and proline present would give you a non-turbid sample, but that is what we see. Additionally, it seems like, alanine octanol is not very cloudy, alanine decanol is very cloudy, and then a combination of alanine octanol decanol is somewhere in the middle. But somehow proline octanol, not cloudy, proline decanol, cloudy, proline with octanol and decanol, very cloudy. Again, not predictive. And then when you mix all four together in a reaction, they form a pretty substantial gel phase and the most cloudy solution. And obviously that's not concentration dependent here. This is actually a product of the interactions between, like this is something to do with the interactions between our amphiphiles. And so I think that what this tells you, even in this 4-part mixture, is that there are things that are non-predictive about structure formation of prebiotic amphiphiles that you can't necessarily look at how much product you're making or how much abundance there is and determine whether or not vesicles will be present. And that complex mixtures are not something that has been well tackled by the astrobiology community to date, especially the prebiotic chemists. So with that in mind, I have been funded for an NSF Molecular Foundations of Biotechnology grant, where we're going to try to use machine learning to predict lipid formulations for the origins of life. We have funding for 2 postdocs. My collaborator, Joshua Schreier at Fordham, which is in New York City, is looking for someone to work on the machine learning side of things. He essentially says, if you have a PhD and you know something about computer programming, you're probably qualified for this position. And I am looking for someone who kind of likes astrobiology and wants to do some analytical chemistry and wants to work with undergraduates. And that's going to be a key part of my postdoc program. And so if you're interested or you know someone that's interested, I will be hiring sometime in spring, hopefully. Okay. So I'm just going to talk a little bit more about realistic chemical mixtures for the second half of my talk. And I'm supposed to tell a joke first. So you want to hear a joke? Intermission. This is intermission. All right. Why can't you mix a bear from the Arctic with a bear from Montana? One's polar and one is non-polar. All right. Okay, great job, everyone. Okay, so I have spent some time thinking about realistic chemical mixtures, and this is not a protocell talk at all, although I do still have an interest in phase separations in these mixtures. I think that we can use the idea of aggregation more broadly and still think of it as kind of a organizing system, much like a membrane would be. So I talked a little bit at the beginning about these high energy radiation reactions that occur on meteorites or in terrestrial synthesis in spark discharge. When you actually analyze spark discharge in a mass spec, each one of these vertical lines represents an individual mass. And inside of each one of those vertical lines, you could have many different isomers. So assembling the carbon, nitrogen, hydrogen in different ways so that you get essentially different molecules at each mass. And so from a single spark discharge experiment that is starting with, essentially 3 reactants and a bunch of energy, you end up with hundreds of thousands of products. And somehow we are proposing as astrobiologists that hundreds of thousands of products go from being very molecularly diverse to being very molecular functional and limiting that molecular diversity, right? So that's the hard problem in prebiotic chemistry. You can start with a binary mixture You can also start with a quaternary mixture, but you can also start with a actual realistic chemical mixture. I don't have a spark discharge experiment in my lab. Jim Cleaves was like, oh, just build one. And I was like, I don't think you know what I do. There's no chance of me building one. And so instead, I asked students to mix 100 chemicals that are found in meteorites together. And we called it a synthetic prebiotic soup. So it includes amines, hydroxy acids, amino acids, fatty acids, aldehydes and ketones. We put in sulfate and carbonate and other things you would expect from meteorite losses. And from these, it was like maybe more than 100 different compounds. We put it through some kind of what I call chemical evolution. And what we mean by that is we take a fixed set of small molecules, shown here as 3, really hundreds, and we put them in different environments. And in this example, we're using wet-dry cycling, but I am currently working on using UV light, redox chemistry, specifically different iron sources, and a little bit of hydrogen peroxide to kind of drive these environmental changes in our 100 molecule set. And then we can get out different metrics of evolved. So you can use mass spec to quantify how evolved your system is, but you can also use things like how functional your outputs are, or you can use things like what they look like under the microscope, how turbid they are. Do they interact with specific other molecules, right? So looking at interactions, like, do they interact with RNA, would be an example of a evolution. And we can compare our starting mixture to our evolved mixture. And the difference would be how evolved the system is. And when you're working with really big data sets, really big chemical mixtures, you cannot quantify every single product, and you cannot quantify every single loss of reactant, right? It's just not possible. It may not even make sense, but analytically it is very, very challenging. And so the best bet we can do is to get a bunch of different readouts from this. So for example, what are the different functions, or, how many peaks do we see between 100 and 200 mass units, and use those as metrics for the evolution. And then we can reduce the dimensionality of that data. So in this data set, I took 100 different molecules. We put it through a few different assays and a few different mass spec readouts. And we had a 12 by 12 matrix of features of our evolved system. And then we ran principal component analysis. If you're not familiar with multivariate statistical analysis, essentially it looks at where the biggest deviations are in your sample set, and then it tries to group your samples. And so the samples here are grouped by so they all started with the same organic compounds, except this very big red grouping was 3 times the concentration of those organics as compared to the green grouping and the blue grouping. So the concentration of the organics that you put in at the start controls how they differentiate, right? So having more of your reactants is going to have a different grouping than having less. So it is important what your starting concentrations are. Additionally, if I took my organic soup and I added an oil droplet to it, it also differentiated. So I just added 10 microliters of mineral oil to the top of my sample, and it has a different set of outputs from my non-mineral oil containing samples. So we do think that having these aggregated surfaces is going to play a role in how chemicals evolve. Additionally, you can take these subsets and look at their differentiation as well. We find that if you're rehydrating with pure water, that we're seeing differentiation from rehydrating with more soup, so like a dilute soup, or this really big purple circle is dilute soup plus seawater. And so adding different environmental factors when you're rehydrating is going to, of course, drive your evolution. And then finally, even if you're just rehydrating with pure water every single time, depending on the amount of salinity in our samples, so the red group is low salinity and the green group is ocean salinity, and the blue group is 2 times the modern ocean salinity. Again, we get differentiation. So we started with the exact same organic inputs. We just change the salinity of the sample, and we see that we get different evolutionary outputs, or the chemicals evolve differently depending on the environment. And so we know that we are going to get different products depending on, our pH, our salinity. I was surprised how strongly this differentiates with salinity, because you would think that pH would be a larger driving factor, and it did not seem to be. They differentiated better from salinity. We assume that also temperature can play a role in this differentiation. So we haven't done it yet, but controlling the temperature that you're dehydrating at likely drives different product formation. I'm sure a number of cycles does too. And like I said, I'm now trying this with some other energy inputs, so not just doing wet dry cycling. Wet dry cycling is really popular in prebiotic chemistry right now, which is why it's like a low-hanging fruit. Also, it's really convenient because undergrads like come in on a Tuesday and then a week later they rehydrate on a Tuesday. And so they have like this really nice work schedule. The other thing that we found is that these soups evolved functionality differently depending on their environment. And this is kind of hard to imagine, but we're looking at 3 different measures of functionality. So in our first reaction, we're looking at our soup's ability to reduce copper 2 into copper 1. We've also looked at our ability of our soup to cleave a ester bond. This product, p-nitrophenol, is yellow, but the reactant is not yellow. So this is really easy to do visually. And then we also can attach that p-nitrophenol to a phosphate and look at phosphate bond cleavage. And these are really common biochemical assays that we're applying to a mixture of 100 different compounds that has been evolved. And what we see is that depending on, for example, with the reduction of copper, depending on the acidity of the sample, we see better ability to reduce copper or a worse ability to reduce copper. So the organic mixtures that were evolved at pH 10 were not as good at reducing copper as the samples that were evolved at pH 2. And when I did this, we of course adjusted the pH to pH 7 for the assay, or to its high pH because it's the BCA assay for the assay. And similarly, esterase activity seemed to be controlled a little bit by pH, but really interestingly, phosphatase activity, you would think that, you're cleaving off this same p-nitrophenol group. And so if this is driven by the pH, then why wouldn't the phosphatase activity be driven by pH? But it turns out the phosphatase activity was driven by the salinity of the sample. That's what controlled whether or not the products were able to cleave that phosphoester bond. And so we're getting differentiated enzymatic activity here based on the environment that our organics are evolved in, which is just really, really unexpected. I obviously have interest in making more of these assays for some of our big grant because they're very convenient to do high throughput screening on. The other thing that I'm very interested in, and that the astrobiology community might be interested in too, is that I have a kind of 1-year seed funding from Heising-Simons Foundation to look at how we can model the oceans of Enceladus and do a very similar reaction where we look at how primitive organics are evolved in the conditions that we see, or that we estimate are in this subsurface ocean of Enceladus. I'm sure I have to say this, it's one of the moons of Saturn. And then also think about how those chemistries enter into plumes and compare it to what we see from both Cassini data and from telescope data. And so I'm going to work on making a actual physical mixture in the lab that resembles Enceladus's ocean. We very crude in my lab. And so if you have ideas on that, I'd love to talk about it. And we are also interested in then taking feedback. So if you have plume data, how can we incorporate that into our model better? And I think that this is a really interesting way to think about what a false signal will look like, because we know that we can evolve functionality. And so can we evolve things from the soup that look very interesting, but are not actually biology, because I made them in my lab and I'm the only biology that's in there. All right, so I hope that I have interested you with all of my various products, from our inclusivity ideas, the idea that you can make archaeal membranes. Oh, I didn't really talk about that. We're trying to make liposomes out of archaeal extracts. So if you have archaea, I would love to take some of it and see if I can make liposomes out of it. We also are interested in how prebiotic vesicles are formed from amphiphile synthesis all the way up to characterizing their ability to be cell-like, and looking at complex mixtures and exoplanet modeling. I know this is, like I said, I have many hands in many pots. These are my undergraduate students. Adam is now at Northeastern, Kiernan is at University of Virginia, and Liz is looking for a home. And so is Mia. And so I have a pretty good group of students that are currently working with me, or have worked with me in the last year and a half. Like I said, turnover is really, really high. This is like a year and a half of students. And so, yeah, I think I'm always looking for collaborators and opportunities. So please, if you have any questions.
Okay, I'm sure Sarah would be happy to take questions. So does anybody have any questions? Roy.
So your esters, amino acid, decyl esters that form the vesicle. The one thing that I wondered about with that kind of molecule is how stable is, I mean, I always had the idea that esters aren't that stable when you get at neutral or much above. So how stable are these things, and where have you done most of your experiments? The microscopy, was that at the acidic pH at which they were formed, or?
Yeah, so for the online crowd, Roy just asked how stable my esters are. And if you're not a chemist, esters hydrolyze at high pH. And so that's the nature of the question. And so if these are prebiotic amphiphiles, how robust are they? And I think this actually is a unique system. And so, yes, at pH greater than 10, they do hydrolyze fairly rapidly. Days is best estimate. It probably depends a lot on your concentration and how much you have left. Below 10 though, so you can bring the pH up to 9, as long as you don't deprotonate that head group, they're very stable. So we're talking about, you know, pH 1 to pH 9 as a range for these products. The other thing that I would like to do in the near future is see if the molecules that form membranes degrade more slowly than the molecules that form micelles or don't form membranes. And so, like, what if high pH was actually a selection mechanism for membrane formers, and you could then selectively degrade the amino acid esters to like reuse those amino acids for proteins, for example. And so it could be you could torture a system enough that it would be a benefit.
And one more, and then I won't not really. Just what about the integrity of these vesicles in terms of keeping a guide inside? How leaky are they?
So we have not attempted to encapsulate solutes or optimize their formation in any way. We literally are just rehydrating with 5 ml quantities of water and characterizing turbidity and morphology. I think longer term, that may be something that we are interested in. I think one of the things that might be happening in our samples is that there's still some portion of unreacted decanol, and there's probably still some portion of unreacted amino acid. And so it could be possible that those things are stabilizing some of our membranes and also destabilizing some of our other membranes. So we have attempted to purify some of this, and once we have more purified samples, then we can do things like encapsulation.
Online.
Josh, do you want to go?
Hi, yeah, I thank you for that talk. It was really, really cool. So just to give you a perspective of where my question is coming from is I work on metabolomics, primarily in marine environments. So thinking also a lot about complex mixtures. And I was wondering what sorts of like analytical approaches you have been applying and that you're considering applying to your interpretation of those complex mixtures. Because it almost seems like a lot of the tools of the metabolomics world or the dissolved organic matter world would be really useful for you. And so in terms of combining your mass spectrometry with gas chromatography or liquid chromatography, in addition to like the software like that's available for identifying exact masses and predicting structures or molecular formulas for compounds you're interested in. So I guess I was curious how you were planning on continuing to explore those, because it's really, really cool work.
Yeah, I think that that's very, very spot on, that there are a lot of tools outside of what I've been using that could be very, very helpful in better understanding our systems. I don't have that expertise, nor do I know anyone with that expertise. Actually, when I was on sabbatical at the University of at Georgia Tech, I talked to the metabolomics people, and we thought about different ways to collaborate, and it just didn't come to fruition. Right now, what we're doing is really tediously by hand, making sure that our that our UV data is consistent with itself. We do everything in triplicate and make sure that like baselines are always at 0, for example, by hand. My student did not love this process. And then we were looking at total peak areas in specific time ranges without any idea of what specifically was in that time range. I mean, we know things that elute early in a C18 column are usually polar, and things that elute late are usually non-polar. And so we had those kinds of metrics, but essentially we just took as many different time ranges as we could, and things that had deviation across samples are things that we used. If all the samples were exactly the same, then we didn't use that data for our statistical analysis. So like very rudimentary, I think, yeah.
Okay, more questions? Guess I have a question, which is, so you kind of dissed the fatty acid decyl membranes in favor of these amino decyl ester things. But is there any reason to believe that, you know, eventually you have to get to phospholipids, which the membranes used by life? And although there's diversity, it's still a phospholipid. Is there any reason to think that this direction would take you there in a better way than starting with fatty acids? Because eventually it seems to me that what you need to do is add a glycerol phosphate group onto these, somehow get that to meld onto these things, which is not necessarily obvious why these ones should be better at that than the fatty acids.
I'm just going to ask, did everyone online hear that question? Because it was long and I don't want to repeat it. Okay, cool. We're going to
Yes.
yes. Okay, so the answer is that because lipid diversity is really, really high, we think that there is not a strong selection for specific membrane formers. But on top of that, it is very possible that lipid biosynthesis came later than the other biosyntheses that we see. So we first had protein and nucleic acid and sugar biosynthesis, metabolic intermediate biosynthesis metabolism, and that lipids essentially were just scavenged from the environment because it's very easy to aggregate hydrophobic material. And so picking a specific lipid would be less important than the ability to incorporate existing lipids into your membranes. I don't know that that's entirely true. It could be that you needed the ability to synthesize lipids directly to have any sort of life. But because lipid diversity is just, it is really, really so high. And we don't need glycerol esters necessarily because just because we see that in both archaea, bacteria, and eukaryotes. I mean, we know that fatty acids form membranes. We know that alcohols can contribute to membrane formation. And, you know, cholesterol is essentially an alcohol. And so I guess I'm less focused on getting the explicit building block that life uses today and more focused on building a robust membrane that can support early chemical systems.
Okay. So I have another question, which is naive, but what do you really mean by chemical evolution? Because the term is bandied about a lot, and it seems to mean chemical changes that happen and may go in certain directions depending on the environment. But where do you hope to end up in this in order to get to something that enables prebiotic chemistry and the origin of life? What's the metrics that you use?
Yeah, I think that the word complexity is bandied about in astrobiology in a lot of different ways. And I would caution young astrobiologists to use that term very carefully, because a lot of times when chemists are talking about complexity, what they mean is high molecular diversity. It's a mixture that I can't analyze. Whereas, you know, when we think about evolutionary complexity, it means that there are kind of hidden substructures in our network that are more complex than just, you know, having a peptide, right? And so when we think about chemical evolution, what we are evolving for is less overall molecular diversity, right? We don't want to have 100,000 different molecules, but the molecules that we do have, we want to be more chemically functional. And functional in this case could mean the ability to co-survive. So like the interaction between two molecules to just survive specific conditions. But it could be the ability to catalyze reactions. It could be the ability to help in membrane formation, right? Or to like catalyze membrane formation, or to increase membrane permeability, or to, I mean, I think a lot about membranes, so I'm going to give you like 30 membrane examples. But essentially that the evolution reduces the molecular diversity, selects for some kind of functionality.
Okay, thanks. Okay, any more questions? If not, let's thank Sarah once again.